Hello, my name is Pia Lange, and I present to you my poster about exploring DEG calling in the presence of limited patient samples. DEGs are genes that are differentially expressed in two or more conditions. Here we have expression data for condition A and condition B. In some cases, it is very clearly to identify the DEGs, for example, gene 1, but sometimes it is harder when we have outlier samples as B3 for gene 2 and a very high variance as in G3. The obstacle here is mainly that we only have 3 samples per group, which makes it very hard to estimate the true mean and variance and to identify the outliers. It would be nicer to have more samples, but in reality, researchers can only offer a certain amount of samples. Reasons could be difficulty in finding patients or financial limits. From my experience, requests for analyses with less than 5 samples per group are very common, so I wanted to explore whether those kinds of analyses are feasible. To estimate the performance, we needed data for which a ground truth was present, so we simulated synthetic data with the comcode RR package, which uses a negative binomial distribution with mean and variance taken from real data to randomly generate expression matrices. We tested sample sizes ranging from 2 to 10, and the matrices had a total number of 10,000 genes per sample size. We defined 4 distinct cases where either 5, 10, 30, or 50% of genes were truly differentially expressed. The size of the difference was taken from an exponential distribution with expected values ranging from 1.5 to 2.5. To include the possibility of errors, each count had a 5% chance of becoming an outlier. For our analyses, we used the tool DEG2. It assumes a negative binomial distribution and then estimates dispersion and effect size based on the mean and variance of the data. We used this tool because benchmarking literature showed that it is relatively sensitive in the presence of only a few samples compared to other tools. And it has some interesting features that are worth looking into that specifically help when working with a low amount of samples, such as outlier flagging and replacement and global dispersion estimate, also called shrinkage, instead of local dispersion estimates. Now I will show you the results of our setup. On the top left, we have the results for 5% DEGs, then 10, 30, and 50. On the x-axis are the group sizes. In blue, we see the recall of the true positives. Generally, we observe that recall steadily increases along with the group size. However, for the chosen group sizes below 10, we barely reach 40% recall. In red, we have precision, so the percentage of true positives among our results. And even for a few samples, relatively high precision can be retained depending on the fraction of true DEGs. For example, here for 30% DEGs, we only need 4 to 5 samples regarding precision, whereas for 5%, we would need 8 to gain maximum precision. The percentage of the true DEGs seems to be a major factor for precision in general, and researchers should be aware that analyzing very similar conditions, like the top row, might be prone to errors as we do not exceed 75% precision here. So sometimes samples are divided unequally among the conditions. For example, we could have a very small pool of patients with a rare condition, but a large reference group of healthy patients. Here we have tested a group size of 10 as some kind of best case comparison. And on the right, we have 3 samples per group. Now we assumed that for one of the groups, samples are easier to acquire, so we increased one of the groups size to 10. This only moderately increased recall and did not increase precision at all. If we instead increase both groups to 5 samples, we have a similar increase in recall and a larger increase of precision. Therefore, it might be better to evenly distribute the samples. As recall is the limit of small group sizes, researchers should not hope to gain extensive insight over all differentially expressed genes and networks. Instead, one can search for candidate genes to investigate later. To avoid select false positives here, DEGs should be re-evaluated. For example, this is done by ranking them by effect size. Another strategy we propose here is re-evaluating called genes by intersecting them with results from data subsets. For example, if we had original data size of n equals 4 samples, we do our analyses and then we save our results, and then we choose a subset of n minus 1 equals 3 samples, do a second analysis, and then intersect the new results with our original results. On the right, we have a figure that shows the comparison of our original results, for example, n equals 4, with the intersection results that are shown in the lighter colors. Naturally, we have a decrease in recall, but on the other hand, we have an increase in precision. This strategy could be repeated with another subset of m-1 samples, basically like jackknifing resampling, to further improve precision. Since, especially for small group sizes, results are quite unstable in the presence of outliers and variants, this method could help exclude genes that are only called in the presence of a certain sample. So, at last, let's conclude some tips for working with limited data. First, consider the level of difference between conditions. Comparisons such as cancer versus control with more than 30% DEGs should be safe, whereas be careful with comparisons of smaller with similar conditions. Then one should probably take additional steps to increase recall, such as filtering low expression genes. And when increasing group size, focus on increasing the size of the smaller group if possible. At last, it is safer to aim for explorative research to generate hypotheses instead of hoping to get an extensive overview of all DEGs because of the low recall. So, thank you for watching my presentation and feel free to contact me if you have any questions.